Good morning. It's September 22nd, and this is your daily brief in science. Here's everything you need to know. gc-biopharma has submitted a domestic IND application for its COVID-19 mRNA vaccine candidate, gc-4006A, to the Korean Ministry of Food and Drug Safety. The goal is to evaluate its safety and immunogenicity in healthy adults. The company announced on September 22nd that it is moving forward with clinical trial plans for gc-4006A, which was developed on an in-house mRNA LNP platform since 2019. Development of gc-4006A was accelerated after the company was selected for the Korea CDC's Pandemic Preparedness mRNA Vaccine Development Support Project. This project aims to localize mRNA vaccine production in Korea by 2028. The initiative has helped gc-biopharma advance its research and development efforts, supporting local vaccine manufacturing and research through 2028. The company also plans to expand its mRNA platform to other therapeutic areas, including rare genetic diseases, cancer immunotherapy, and inflammatory conditions. gc-biopharma's proprietary mRNA LNP platform enables it to handle the entire development process independently. The company aims to obtain IND approval within 2025 and start clinical trials early next year. The platform's versatility could allow for rapid responses to future pandemics within 100 days. In non-clinical trials, gc-4006A has shown the ability to induce both humoral and cellular immune responses comparable to existing vaccines. These results indicate promising efficacy. Overall, gc-biopharma's efforts reflect a strategic push toward a self-reliant, versatile mRNA vaccine platform capable of addressing current and future infectious diseases. Researchers at the University of Osaka have developed a minimally invasive brain recording technique that uses ultra-thin wire electrodes inserted into cortical and deep cerebral veins via a catheter. This method achieves high-fidelity recordings comparable to traditional invasive techniques. The approach has successfully captured brainwaves from both superficial and deep brain regions in pig models, enabling precise monitoring of brain activity. Additionally, the technique stimulates the motor cortex through these blood vessel-based electrodes, evoking muscle responses in the face and shoulders. This demonstrates its potential for functional brain mapping. By providing access to deep brain regions without invasive surgery, the method opens new possibilities for brain monitoring. It is especially relevant for areas that were previously difficult to reach non-invasively. Led by Professor Takufumi Yanagisawa and Dr. Takamitsu Iwata, the research was published in the journal Advanced Intelligent Systems. The breakthrough could improve diagnosis and treatment of neurological conditions like epilepsy and enhance brain-computer interface technology for individuals with severe paralysis. Dr. Iwata highlighted that this approach might offer a less invasive alternative to traditional brain surgery. The technique's ability to detect brainwaves from deep regions and evoke muscle responses shows its broad applicability, including resting state EEG, somatosensory, and visual evoked potentials. Adding targeted immunotherapy to standard treatment is showing promising results in multiple myeloma. Recent data from the Phase 3 AURIGA trial indicates that combining subcutaneous daratumumab with lenalidomide maintenance significantly increases the rate of minimal residual disease, or MRD, negativity after stem cell transplant. This effect was especially notable in patients who were MRD positive at baseline. The study found that patients receiving daratumumab achieved about a 60.6% MRD negative conversion rate at a sensitivity of 10 to the negative 5, compared to 28.7% with lenalidomide alone. Over time, these rates improved, and the durability of disease control was better with daratumumab, with more than twice the rate of sustained MRD negativity for at least 6 months, and nearly 4 times for a year or more. Progression-free survival was also improved, with median PFS not reached in the daratumumab group versus 47.2 months in the control group, and 3-year PFS rates of 76.8% versus 61.4%. The safety profile was comparable between groups, with no new safety concerns, and adverse events like neutropenia and infections were similar. Patients tolerated the treatment well, with lower discontinuation rates and longer treatment durations observed in the daratumumab group. These findings suggest that incorporating daratumumab into post-transplant maintenance could become a new standard of care for MRD-positive myeloma patients, potentially improving long-term outcomes. Scientists on Nantucket are developing a genetic approach to combat Lyme disease using CRISPR technology. They are inserting an antibody gene into mouse embryos to make the mice immune and capable of passing this immunity to their offspring. This process involves injecting CRISPR and antibody genes into the embryos to create Lyme-immune mice, targeting the primary hosts of Lyme bacteria. The goal is to reduce the population of infected mice, which are key in the transmission cycle of Lyme disease. Community concerns focus on ecological impacts and potential disruption of natural ecosystems, leading to calls for regulatory approval and extensive ecological testing before any large-scale release. The project also raises ethical questions about altering natural evolution and ecological balance. Supporters argue that lowering Lyme disease cases could provide significant public health benefits, potentially reducing reliance on traditional treatments. Initial small-scale trials are planned on a private island, with future releases on Nantucket contingent on regulatory approval and community support. Researchers are engaging with residents to gather input and address ecological concerns. Experts emphasize the importance of thorough testing to prevent unintended ecological consequences, especially considering historical deer introductions that increased tick populations and the role of white-footed mice as key hosts. The scientists plan to release thousands of genetically engineered mice during winter, when native populations are low, to monitor ecological impacts. While there is cautious optimism, concerns about potential effects on predators like hawks persist, underscoring the need for comprehensive ecological assessment. The overarching aim is to decrease Lyme disease transmission by reducing the number of immune mice, thereby lowering the risk to humans. Scientists from IIT-BHU and Banaras Hindu University have been recognized among the top researchers worldwide, highlighting their significant global impact. This recognition is based on research metrics such as citations, H-index, co-authorship, and the C-score. A total of 46 faculty members from IIT-BHU and 44 from BHU are featured on Stanford University's Top 2% Scientists list for 2024. Among these, 44 BHU faculty members are ranked among the top 2% most cited scientists worldwide for their career-long contributions. Additionally, 51 BHU scientists received recognition based on citations in 2024. Professor Amit Patra, director of IIT-BHU, expressed pride in the achievement, emphasizing it as a testament to the institute's research strength and international stature. The recognition underscores the dedication and innovation of the faculty at both institutions. Stanford's list categorizes these scientists into 22 main fields and 174 subfields, reflecting their influence across diverse scientific domains. This annual ranking is updated each year to recognize scientists making a significant impact in their fields. The achievement not only enhances the reputation of IIT-BHU and BHU, but also aligns with their goals of fostering global collaborations and impactful research. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.